Welcome everyone. Thank you very much for calling in today. We are very fortunate to be joined by scientists across the country and the globe. The subject that brings us together today is Achieving API Development Goals Through Solid-State Forms. This webinar is provided by Alcami Corporation. Alcami is a US-based company with headquarters in Wilmington, North Carolina. It is a leading provider in analytical, formulation, and manufacturing services for oral solids and parenteral dosages. Alcami has recently extended the support to earlier preclinical programs by adding new lab facilities focusing on solid-state characterization, solid form screening and selection, and early developability activities. My name is Joanna Biss, and I lead the solid-state characterization operation in Research Triangle Park, North Carolina. Prior to joining Alcami in 2022, I spent 12 years at Catalan Pharma Solutions, managing solid form screen and selection programs. Before that, I worked at GlaxoSmithKline, where I supported late-stage development compounds in the context of intellectual property and product line extensions. I received my graduate degree in crystal engineering of organic molecules from University of South Florida. This webinar will last about 1 hour. I will start with the description of the various solid-state forms and why is it important to select a suitable one for the development. Then I will highlight some of the key considerations important in the discovery of salt polymorphs and cocrystals. I will also share published case studies describing how the various forms were evaluated and how the accumulated knowledge impacted their selection. Finally, I will discuss the tools utilized in the solid-state screening and characterization. Solid APIs can be divided into two main classes, amorphous and crystalline. Amorphous solids do not possess long-range order at the molecular level. The molecules are randomly oriented with no defined patterns. These high-energy materials can exhibit useful properties such as higher exposure and can be developed, but they also pose quite a few risks. Disordered structure lead to variable behavior, physical and chemical instability, and hygroscopicity, and those properties cause difficulties on manufacturing operations such as isolation or purification. Crystalline solids, in contrast, exhibit ordered structure with specific patterns that are reproducible in 3 dimensions, and that leads to well-defined and often controllable properties. Crystalline solids can be further divided into polymorphs, solvates, salts, and cocrystals. Polymorphs exist when API molecules differ in their arrangement in the crystal structure. In the case of a solvate, solvent molecules are incorporated inside of the crystal. Solvate can be organic, such as ethanol solvate. Otherwise, if water is the solvent is called a hydrate. Cocrystals are analogous to solvates with the difference that all components are solids at room temperature when in pure state. Salts are ionic compounds consisting of ionizable API and counterion. Due to the proton transfer between the components, an ionic bond is formed, which is often responsible for significantly impacting properties of the API. In summary, the same API can exist in a variety of solid-state forms, and each of these forms often have specific set of properties. This webinar focuses on crystalline forms of the API. Why are solid forms important? For example, creating a salt of a poorly soluble API can lead to improvement of its solubility profile. An increase in solubility or the solution rate increases the amount of drug that gets into the bloodstream and ultimately to the therapeutic target. Other properties that can be influenced includes physical and chemical stability, hygroscopicity, and handling properties. That interrelationship of structure of the solid-state form and its property extends to process and product performance. Solid forms influence manufacturability, since they can have varying tendency to reject impurities, different behavior on filtration and drying, and may be more or less amenable to secondary processing such as granulation or micronization. Solid forms impact formulation strategies such as route of administration, dosage forms and strength, and ultimately bioavailability, efficacy, and safety. Regulatory agencies such as FDA and EMEA require that the solid-state form processes of preparing and formulating them are well characterized and controlled. And last but not least, the discovery of novel solid forms can result in new processes that can be patented, providing protection for new or existing drug products in the form of product line extension. In summary, it is important to select a solid form with suitable properties to enhance the likelihood of achieving various development objectives, to increase the speed, to control the cost, and to minimize the risk during the development cycle. A classic example demonstrating the implication of progressing a wrong solid-state form into the market is represented by ritonavir, anti-HIV medicine. This example is a company's worst nightmare due to the unexpected polymorphism. Such phenomenon is sometimes compared to a hurricane because of its uncertainty of if and when it's going to hit and what level of devastation it may cause. The drug was launched in 1996 as an oral solid capsule containing solid form 1. In 1998, when Blockbuster was achieving $250,000 in sales, the medicine started failing the solution specifications. It turns out to be due to new form 2 precipitating in the capsule. Form 2 was more thermodynamically stable with 50% lower solubility, therefore was not achieving the expected bioavailability. The solid formulation was withdrawn from the market, putting patients at risk and causing huge financial losses. Abbott's team worked around the clock to understand the new Form 2 and to reformulate it into a novel refrigerated softgel capsule. Therefore, it is important to identify polymorphs, understand their properties, especially solubility, regardless of the type of formulation, solid, suspension, or solution. Solid-state forms are generated via deliberate screening efforts conducted at preclinical stages. Majority of screens focus on discovery of new salts and polymorphs. More than half of marketed products are crystalline salts, progressed mainly for the solubility advantage. More than half of APIs exhibit polymorphism, and about 90% of them can exist as either polymorph, solvate, or hydrate. Co-crystal screens have been gaining increased attention in the last years as they expand the development choice beyond the well-established solid forms. They are alternatives to molecules that do not form salts. The literature on the pharmaceutical co-crystals started in early 2000s and was soon followed by multiple patent applications and approvals. 3 early approved drugs, Entresto, Steglarto, and Suclat, exemplify co-crystal compositions with beneficial bioavailability, hygroscopicity profile, and form control respectively. This very activity, discovery of new crystal forms via polymorph salt co-crystal screen, is at the center of the preformulation studies. It is important to efficiently screen for crystal forms, characterize them rapidly, evaluate them in the context of developability criteria, and select a suitable form for the next milestone, such as PK study or formulation development. The first crystallinity and solubility evaluation of the solid form starts once a molecule is synthesized by a medicinal chemist. If the synthesized parent molecule is crystalline and its solubility is adequate to allow animal PK and initial tox studies, no salt screen is needed. If the molecule is not crystalline or insoluble, crystallization or salt screens are performed to identify a solid form that can support these studies. The crystalline parent or crystalline salt will then proceed to a polymorph screen in order to ensure that most stable form is identified. Ideally, the same polymorph is used in all following development stages to avoid delays and cost associated with a later form switch. However, if this is not possible, it is important that the same polymorph used in the toxicity study is also used in first-in-human formulations to avoid bioequivalent studies. Additional round of screening may be needed later around phase 1 to identify process-relevant and metastable forms in order to discharge any risks to crystallization or formulation. And finally, comprehensive screens can be conducted in phase 2 to support final make and obtain a more comprehensive knowledge of the solid form space that may indicate new forms for patent purposes or new indications. Most companies prefer to identify stable salt and its polymorph early in the development to avoid form switches, and also to use the screening data to guide crystallization and formulation development work. Other clients, perhaps due to resource limitation, conduct smaller targeted screens to obtain just enough data to address specific milestones being faced at a given time. As mentioned, salt screens are typically conducted early and are triggered by issues around the parent molecule. Issues may include too low or too high aqueous solubility, chemical instability, purification, or high propensity for polymorphism. Since salt is composed of anion and cation, its formation depends on pKa difference between the acid and the base. The difference of 2 pKa units is generally accepted for salt formation in aqueous solution. However, the FDA relaxed the pKa rule to 1 unit for the purpose of differentiating salts from co-crystals. Counterions are divided into classes based on the established toxicity data. Some counterions are specifically avoided in screens, for example oxalic acid, which may lead to formation of kidney stones. Animal acceptability is a factor as well. For example, maleate salts are known to form nephrotoxicity in dogs, therefore they should not be studied in those species. Route of administration is also important. For example, injectable formulations accept a narrower range of counterions as compared to the oral formulations. Injectable formulations require that salt's natural pH is in a range suitable for injection. Therefore, salts of very strong acids or very strong bases are not used. While pH adjustment is possible, the salt still needs to be stable and do not dissociate into the parent upon the pH shift. Molecular weight of counterion is considered, usually for high dosage forms for which smaller counterions are preferred. Chemical compatibility is important in avoiding impurities, such as sulfonate esters, which are genotoxic. Those can be formed between sulfonic acid and small alcohols that are used in the screening studies. Therefore, for sulfonate salts, the ester content should always be monitored. Classic sources for counterion selection are the green and orange books, which describe the commonly used counterions in detail in terms of safety, solubility, and precedence in the marketed salts. For orally administered APIs, the need for salt formation can also be assessed by considering biopharmaceutical and developability classification systems. Both BCS and DCS systems consider anticipated dose, permeability, and solubility. The BCS is focused on aqueous solubility and is used by regulatory agencies to grant biowaivers for immediate release formulations. The DCS considers solubility in fasted simulated intestinal fluid and allow for early evaluation of candidates for oral delivery, as well as for more granular understanding of the root cause of the solubility limitations, whether driven by the solution rate or intrinsic solubility. This information in turn can be used to guide design of optimized formulations rather than relying on trial and error. In both classifications, APIs are divided into 4 classes. Class 1 includes high solubility and high permeability, both of which allow for simple formulations such as powder in bottle. Class 3 covers high solubility and low permeability, which allows for simpler formulations with permeability enhancers. Classes 1 and 3 of APIs do not require salt formation for the sake of increasing solubility. Class 2 includes low solubility, high permeability. The DCS further divides it into 2 subcategories. A, the solution rate limited solubility, which is usually mitigated by particle size reduction, and B, thermodynamically limited solubility. This quadrant is separated by so-called solubility limited adsorbable dose, or SLAD line. Class 4 covers low solubility and low permeability APIs. Both classes 2 and 4 are the most frequently studied. 90% of small molecule drug candidates are poorly soluble and require more complex enabling formulations, such as halt melt exclusion or spray-rate dispersion. Challenges related to poorly soluble drugs are low and variable exposure. In both cases, salt screen is critical because a new salt with adequate solubility can move the molecule from classes 2B and 4 to class 1, 2A, and 3 to enable simplified formulations. Let's take a look at an example for solid form selection of an API developed for cardiovascular disease. 2 high dosages were considered, 250 mg for oral solid and 50 mg per milliliter for injectable formulation. The API was a weak base with pKa of 4.1, exhibited relatively low melting at 120 degrees Celsius, and high propensity for hydrate formation. A salt screen was conducted and resulted in several candidates, including hydrochloride, mesylate, and sulfate salts. The HCl salt exhibited a complex melting behavior to hydrate and low solubility, which was unsuitable for injection. The mesylate salt had a higher melting and no polymorphs detected in extensive screens. Solubility was high in water, 0.1 molar HCl, and aqueous 5% dextrose, which were suitable for both oral and injectable dosages. The sulfate salt had highest melting and no polymorphs. Its solubility was acceptable in water and 5% dextrose, however low in 0.1 molar HCl. The mesylate and sulfate salts were non-polymorphic, non-hygroscopic, and the only salts that permitted high-dose formulations. Both salts were further evaluated in formulation vehicles, and the sulfate salt exhibited the most favorable solubility in co-solvents. Therefore, the sulfate salt was selected for development, with the mesylate salt considered as a backup. A salt candidate or a parent candidate is progressed to a polymorph screen. One of the key triggers for polymorph screen is the regulatory requirement for NDA applications to demonstrate a good understanding of a polymorphic landscape and control over the target polymorph, and this is to ensure efficacy and safety. Other reasons to identify new polymorphs are usually issues around physical stability, impurity inclusion, or particle and handling attributes. Polymorph screens also delineate starting parameters for crystallization process development, and they also serve as means to secure intellectual property, since polymorphs usually satisfy the patentability criteria: novelty, non-obviousness, and utility. Polymorph screens can be conducted in various ways depending on the goal. Stable form screens search for most thermodynamically stable form, with emphasis on stability and manufacturability. Those screens focus on incorporating diverse solvents, slow and long-term crystallization techniques such as slurries. They're usually conducted at around room temperature, and they take into consideration aspects such as solubility profile, chemical stability, and involve analysis of dry solids. Metastable form screen, on the other hand, is focused on identification of high energy forms, which, even though unstable, they may be formed upon and impact manufacturing operations. Those experiments involve fast and short crystallization techniques. They take amorphous material as starting point to the experiments. They explore wider range of temperatures and typically involve analysis of wet cakes. Hydrate screens are usually conducted when multiple hydrates exist and they pose a risk to production of a desired anhydrous form, or when a hydrate is progressing to the development. Hydrates are generally viable options for development, since water included in the crystal structure is non-toxic. It is therefore important to understand the stability of a hydrate as a function of water activity and temperature. Those screens look at solubility in water and aqueous mixtures and incorporate wide range and very granular water activity levels. Typically, experiments are done at low temperatures where hydrates are more stable, and when salt or cocrystals are being screened, then that type of study looks at the disproportionation of those versions into the parent molecule. In addition, relative humidity and drying studies are also involved. Process relevance screen focuses on identification, evaluation, and assessment of risks, specifically under crystallization process and formulation conditions. Those experiments utilize solvents and crystallization parameters used in those processes. They typically take final route starting material with known impurity profile. Those screens also involve carryover solvents that may be experienced from preceding synthetic steps, and also study impact of residual solvent in the form of spiking. No matter the scope, the overall goal of polymorph screening is to find as many forms as possible in a cost-effective manner with respect to the material and man-hours. Selecting solvents for polymorph screening is a critical step, and maximizing diversity is key. There are tools out there that support a rational screen design, such as principal component analysis and so-called self-organizing maps, referred here as diversity maps. Scandinavian research teams were able to visualize the diversity of 218 organic solvents spanning a wide range of chemical classes. The resulting diversity map was derived from an assignment of 24 descriptors to each solvent studied and combined with available safety information. The authors incorporated 5 real polymorphic screens on the diversity map. Those screens involve ritonavir in blue, piroxicam in green, formoterol fumarate in red, and carbamazepine in yellow and black, respectively. All solvents included in each of these screening studies are shown on the diversity map. From this exercise, one can see that in the first few screens, which are colored, there is a preference for 10 specific solvents. These solvents occupy a relatively small and concentrated areas of the map, as opposed to the last study shown in black, which covers many of the regions of the map. The larger diversity observed in the last screen is expected because the authors deliberately selected solvents using principal component analysis. While solvent selection will often depend on the properties of API and solvents, such as solubility, viscosity, also solvent availability, cost, and safety, solvent diversity should always be targeted, and one can quickly maximize that solvent selection by usage of those various tools described above. An important portion of a detailed polymorphism study is assessment of relative thermodynamic stability of discovered forms. True polymorphs can exist as monotropes and enantiotropes. In monotropic system, one polymorph is more stable than the other at all temperatures. In enantiotropic system, one polymorph is more stable below transition temperature, and the other polymorph is stable above transition temperature. For hydrates, the thermodynamic stability at a given temperature is a function of water activity and can be established via ripening of forms in specific aqueous solvent mixtures. From those studies, a phase diagram can be constructed that shows the regions of stability for the anhydrate and the hydrates. Both temperature and water activity dependencies are important and need to be considered at all manufacturing operations, including crystallization, drying, storage, and any secondary process that involves relevant temperatures or aqueous conditions, such as granulation. This is to ensure that they operate within the ranges that overlap with the stability region of a target polymorph. For instance, if in the anhydrate, monohydrate, dihydrate system that is shown to the right-hand side, if the anhydrate is desired, it needs to be crystallized or formulated in solvents at water activity below 0.5. And it also needs to be stored at low humidity conditions in order to avoid conversion to the hydrates. A good example of a polymorph screen is one conducted on delta-9-THC naphthol ester, which is an intermediate used in the synthesis of the 9-THC. 3 non-solvated crystal forms were reported, forms A, B, and C, with form B believed as the most stable polymorph above -15°C. Crystallization process of form B was already in place, however it could not achieve satisfactory purification of form B from a stubborn impurity. So client approached us with the task of conducting a screen of the ester to confirm that 1. The progressed form B is the most stable, and 2. To assess rejection of the undesired impurity under various crystallization conditions. The comprehensive form screen was conducted and consisted of 550 experiments and involved 140 unique solvents. In addition, 2 input materials were used, forms A and B, and 5 crystallization modes and temperatures between 5 and 60 degrees C. The screen resulted in the 3 previously known non-solvated crystal forms A, B, and C, a class of 6 structurally similar solvates, a transient form, form F, and new 3 non-solvated forms, forms E, G, and H. Even though form B was the predominant product of the polymorph screening, it was determined via competitive ripenings to be metastable. Its conversion to more stable forms was very slow, taking up to a month of stirring at various conditions. Its slow conversion was probably due to low solubility, potentially present impurities, or slow nucleation and growth of the more stable forms. Competitive ripening of all non-solvated forms A, B, C, E, G, H, conducted at various temperatures, confirmed that between 25 and 60 degrees Celsius, group H is the thermodynamically stable form. At 5 degrees C, experiments were incomplete within a month. However, they also suggested that group H is more stable. Therefore, it was recommended that group H is the progressed form for development. Therefore, the program needed a form switch. This led to the redesign of a current crystallization process. In the next phase, the study explored assessment of the depletion of the undesired XO impurity by the various polymorphs and under various crystallization conditions. The target was to obtain higher impurity purging level as compared to form B, which was at 50%, ideally above 70%, while maintaining reasonable yields of above 80%, as well as stable polymorph. The work involved 120 slurry experiments, a range of solvent mixtures, ratios, volumes, and isolation temperatures. The result of the purging study indicated that the exfoliation was inversely proportional to the yield. Depletion of the most stable form H and some of other solvates were at higher levels as compared to those of form B. For instance, crystallization of form H from 5 volumes of toluene ethanol in 1:9 ratio landed at 70% depletion and 90% yield. Another example, crystallization of DMSO solvate from 40 volumes of DMSO methanol in 9:1 ratio resulted in 77% depletion of the impurity and yield of 80%. The purging study also showed that depletion increases as solvent volume and isolation temperature increases. However, both factors led to lower yields. Therefore, the acceptable combination of impurity level and yield in the context of manufacturing costs needed to be considered. This work illustrates the value of conducting well-designed systematic polymorph studies to ensure form control and impurity control on downstream operations. Let's now talk about pharmaceutical cocrystals, which are a viable development option, especially for non-ionizable molecules which are not feasible to make salts. However, both ionizable and salt products can also be co-crystallized. Co-crystals provide solubility advantage usually in the form of spring and parachute effects, which are similar to those observed in amorphous dispersions. This effect provides a window of sufficient bioavailability within a given time frame. In fact, this was the case for a successful development and approval of Entresto from Novartis mentioned earlier, the first multi-drug co-crystal composed of 6 molecules of succubitril, 6 valsatran sodium salts, and 15 molecules of water. The pKa rule is considered as well for co-crystallization, however to some extent. As mentioned earlier, the pKa rule of 2 or 1, which is used in salt preparation, is applicable to aqueous solutions only, not necessarily solid state. Studies of 6,500 acid-base crystalline structure reported that ionization happens when delta pKa is 4 or greater. For delta pKa of -1 or less, neutral complexes exist. Between these two values, -1 and 4, the proton transfer cannot easily be predicted. Similarly to salt formers, the suitable co-crystal formers can be selected from previously mentioned green and orange books, as well as from the FDA-approved list of compounds that are generally regarded as safe, or everything added to food in the United States. Most recently, natural compounds or nutraceuticals have also been reported in co-crystallization studies. The success of co-crystal preparation is based on principles delineated in the 90s by Ether and Desiraju, namely hydrogen bonding rules and supramolecular syntons. These concepts establish that it is possible to design and create co-crystal structures based on predictable motifs sustained by strong, directional, and reproducible hydrogen bonds. These concepts are the essence of modern crystal engineering, which establishes and utilizes the understanding of intermolecular interactions in generation of novel, multi-component crystal structures with useful properties. Extensive studies within the field led to the generation of synton hierarchies. Two categories of supramolecular syntons were defined: homosyntons and heterosyntons. Homosyntons are composed of self-complementary functional groups such as carboxylic acid homodimer. Heterosyntons are composed of different but complementary functional groups, as exemplified by acid pyridine heterodimer. It was found that in co-crystals the formation of heterosyntons is generally favored over homosyntons. Further studies on heterosyntons involve their formation in the presence of other competing functional groups. For instance, in systems where 3 moieties are present, carboxylic acid, pyridine, and primary amide, the carboxyl and the pyridine moiety consistently interact to form a heterosynton, whereas the amide moieties consistently interact to form homosyntons. In another case, where structures were composed of phenyl, pyridine, and nitrile moieties, the phenyl pyridine heterosynton reliably persisted in the co-crystal structures and took priority over the other possible syntons. The consequence of this hierarchy is that it is possible to rationally design cocrystals by selecting cocrystal formers that will interact with high probabilities. Identification of reliable syntons is facilitated by the Cambridge Structural Database and Mercury Materials module. The potential therapeutic value of cannabinoids in the treatment of diseases such as seizures, lack of appetite, pain, inflammation, and so on has stimulated research and development in formulating them into pharmaceutical compositions. Cannabinol, specifically a member of cannabinoid family, is a mildly psychoactive agent found only in trace amount in cannabis plant. CBN exists as an oil in natural state and is not easily filterable. Cannabinoids in general are highly lipophilic and exhibit very low aqueous solubility. They are also susceptible to degradation to light, temperature, and oxygen. Therefore, formulations including crystalline forms of CBN are presumably more advantageous in terms of chemical and physical stability, possibly solubility, hygroscopicity, and processing. Self-formation of CBN is not an option due to lack of ionizable moieties. A client came to us with a task of performing a cocrystal screen to find viable cocrystals for development of CBN. Cocrystal screen was conducted and used the CBN oil as input material, and that screen resulted in several cocrystalline materials including L-proline and tetramethylpyrazine. Both cocrystals were pharmaceutically acceptable and suitable for traditional crystallization from solution. They also could be obtained in high yields and exhibited very high rejection tendency for major impurities. As a new composition of matter with utility, both cocrystals were captured in a patent application. There are many techniques available for identifying new polymorphs, and for that matter salts or cocrystals, and they can be divided into solvent-based methods, which are listed on top with light gray color, and solvent-free methods, which are listed on the bottom with dark gray color. Common methods are mainly solvent-based and include slurry ripening, evaporation, cooling, and anti-solvent addition. These are popular approaches because they can incorporate a wide range of diverse conditions: solvent mixtures, temperatures, rates of cooling or anti-solvent addition or stirring. They also give an early indication of suitable crystallization process for later development. Solution-based methods are also amenable to automation and high throughput. Solvent-based techniques evolved over time into more specialized technologies to deal with more challenging molecules. Those include use of additives, polymers, gels, supercritical fluid lyophilization, and so on. Some of these methods require specialized equipment, but they do expand crystallization space into areas that are not accessible by common methods. Solid-state methods are also commonly used in screens. For example, exposure of solids to water or organic solvent vapors to facilitate conversion to hydrates or other forms. Other solid-state methods such as grinding, heating, melting are commonly used in cocrystal screens or comprehensive polymorph screens. Some solid-state techniques are also used for special purposes, such as compression can be done to monitor for changes in solid state upon micronization or tableting. These are mostly manual techniques and applied to one or handful of samples at a time. Slower or longer term conditions, which are shown on the left side, favor thermodynamically stable forms, whereas faster conditions, shown on the right, favor metastable forms. Early screens usually involve common screening techniques, whereas comprehensive screens, which are done later, use a combination of most of the listed techniques. A typical amount of solid needed per one crystallization experiment requires a few tens of milligrams. However, some very specialized automated platforms can operate at 1 milligram scale or lower. In terms of analytical tools utilized in the solid-state characterization of small molecule APIs at the preclinical level, there are a few major techniques of interest. I will start with microscopy, which is the go-to technique to discern if a sample is crystalline or amorphous. In our laboratory, polarized light microscopy is used as the first pass method to assess if a screen produced crystalline product or not. Simply speaking, an amorphous sample will not exhibit birefringence or double refraction under polarized light, whereas crystalline sample will. PLM can also distinguish crystals based on size and habit, as well as identify single crystals for X-ray diffraction analysis. Next, in the solid-state toolbox is very powerful powder X-ray diffractometer, which we use as primary detection method in the discovery of polymorph salts and cocrystals. PXRD also differentiates crystalline forms from amorphous, but in addition, it generates a characteristic pattern that is used in identification of crystalline forms. PXRD can also be used to assess phase purity of bulk material and is used in batch monitoring to demonstrate form control under various process conditions. Following are thermal methods, namely thermogravimetric analysis and differential scanning calorimetry. They are different but very complementary techniques. They assess thermal behavior of API upon heating. TGA measures weight loss before thermal decomposition that can be due to a hydrate or a solvate, and during thermal decomposition. DSC measures onsets or hits of transitions of key events such as melting, dissolution, polymorphic changes, and decomposition. Spectroscopic methods applicable to solid form screening and characterization that we use in our lab are FT-Raman and FT-IR. These are very useful tools that confirm chemical integrity of the screening products. In addition, the collected spectra are characteristic for a given solid form. Therefore, they can be used as secondary detection method to complement the previously mentioned PXRD. FT-Raman specifically is also amenable for high-throughput analysis. Our favorite hyphenated technique is TGA-IR method, which is used for detection, identification, and quantitation of volatile components present in a solid, all in one short step. Dynamic vapor sorption, or DVS, is another important technique used to assess hygroscopicity of the API. Powders are exposed to relative humidity levels between 0 and 98%, and the change in mass within a given time frame is measured. If API is highly hygroscopic, it will pick up lots of water and become sticky and eventually turn into a liquid. More stable solids may remain free-flowing powders during the analysis. However, as shown in this example, solids may pick up water only above critical RH level, above which they can turn into a hydrate. Hydrate then can be physically stable across wider RH ranges. Hydrates have significant implication in pharmaceutical development as they may affect solubility, they may become amorphous upon drying, or impact formulation strategies. Therefore, it is important to assess hygroscopicity and the tendency to form a hydrate for APIs in early development. Other tools applicable to solid-state characterization include single crystal X-ray diffraction, which can establish atomic connectivity in molecule and describe how molecules pack in the three-dimensional crystal structure. Therefore, this technique is regarded as a definite evidence of polymorphism. This technique can also verify absolute configuration of chiral centers in optically active compounds. Proton NMR is another spectroscopic method that can confirm molecular structure, and therefore it is frequently used in characterization of salts and their stoichiometry. It can also provide insight to presence of solvents as well as purity of the material. Solvate identification can also be achieved using classical gas chromatography method, as well as novel SIFT-MS technology, which is recently introduced by Alcami to solid form screening workflows. This wraps up today's webinar. Thank you very much for your attention.